All right, let's talk about glycine. Now, on the surface, it looks like the simplest amino acid out there. But trust me, you do not want to underestimate this one. We're about to see how this tiny little molecule is actually a huge player in our body's biochemistry, a real multitasking marvel.
I mean, just think about that for a second. The very code of life in your DNA, the heme that carries oxygen in your blood, and the creatine that powers your muscles, they all trace back to glycine. It's a perfect example of how the most basic biochemistry creates the complex functions we rely on every single day.
So let's dive in. To really get a handle on this, I want you to picture glycine as a central hub, kind of like a busy airport for metabolism. It's this one spot where a ton of critical pathways connect. So we're going to explore how this hub gets built, all the jobs it does, and of course, what happens when it breaks down. Okay, first things first, where do we get this stuff? Well, since glycine is a non-essential amino acid, our body can actually make it itself. So let's look at the two main ways we go about building this central hub. Now, the main route, and this is the one you absolutely have to know for your exams, starts with another amino acid, serine. What's really cool here is the teamwork going on. You've got the enzyme serine hydroxymethyltransferase and its critical cofactor, tetrahydrofolate, or THF. This reaction doesn't just give us glycine, it also spits out a one-carbon unit attached to that THF. This is our first big clue about its deep connection to one-carbon metabolism, which is just a fancy way of saying the body is shuffling single carbon atoms around. It's a theme that's going to keep popping up.
There is also a minor pathway, just so you know the full story. The amino acid threonine can be split by an enzyme called threonine aldolase, and that gives you glycine and acetaldehyde. It's not the main production line, for sure, but it's another way our body can add to its glycine supply.
All right, so we've built the hub. Now let's look at all the different roads that lead out from it. These are the spokes of our hub. And this is where you really start to appreciate the incredible versatility of glycine. It's amazing.
First up, a huge structural role. Glycine is by far the most common amino acid in collagen, the protein that basically holds our bodies together. Its tiny size is everything. It's the only amino acid small enough to fit inside the super tight collagen triple helix, letting it wind up properly. Without enough glycine, our connective tissue wouldn't have its strength. It's that important. Now let's talk about blood, the very first step in making heme. That's the molecule that holds the iron in your hemoglobin. It all starts with glycine. Glycine combines with succinyl-CoA, and that kicks off the whole process. It's simple, really. No glycine, no heme, no working hemoglobin. It's a direct can't-do-without-it kind of relationship.
And what about energy for our muscles? Well, glycine provides the original backbone for creatine. Sure, other amino acids chip in pieces later, but it all starts with that glycine frame. This creatine then gets converted into phosphocreatine, which is that readily available energy reserve that powers your muscles for quick, intense bursts of action.
This next one is just wild. When our body builds the purine rings from scratch, the A's and G's in our DNA and RNA, it doesn't just borrow a little piece of glycine. No, it takes the entire glycine molecule and plugs it right into the structure. This just perfectly shows you how glycine is literally one of the fundamental building blocks of our genetic material.
But glycine isn't just for building things. It's also a messenger. In our central nervous system, especially in the spinal cord and brainstem, it's a major inhibitory neurotransmitter. It basically tells neurons to chill out, making them less likely to fire. You can think of it as a natural brake pedal for parts of our nervous system.
So, we've seen how we build glycine and all the incredible things it's used for, but what happens when there's too much of it? The body has to have a way to break it down and recycle its components. So let's see how the hub gets taken apart.
The main way we break down glycine is through something called the glycine cleavage system, or GCS, inside our mitochondria. This big enzyme complex takes glycine and splits it into carbon dioxide and ammonia. But take a look at that third product, N5-N10-methylene THF. Sound familiar? It should. That's the exact same one-carbon unit we saw being made when glycine was synthesized. So breaking down glycine actually puts a one-carbon unit back into the folate pool. It's a perfect, beautiful example of metabolic recycling.
Now for the minor route, but one with some major clinical consequences. Glycine can also be turned into glyoxylate, which then gets oxidized into oxalate. And why should you care about oxalate? Because if you have too much of it, it can team up with calcium in your kidneys and form calcium oxalate stones. Yep, the most common and painful type of kidney stones.
And that brings us right to the clinical side of things. What happens when these metabolic pathways, the machinery of our hub, stop working correctly? Well, as you can imagine, the consequences can be pretty serious.
Let's compare the two main disorders you need to know. First, there's nonketotic hyperglycemia. This happens when that glycine cleavage system we just talked about is broken. Without it, glycine builds up, especially in the brain, causing devastating neurological problems in infants. Then we have primary hyperoxaluria. In this case, a problem with glyoxylate metabolism leads to a massive overproduction of oxalate, which means recurrent, painful kidney stones.
And building on that, here is a super high-yield fact for your exams. A key diagnostic clue for non-ketotic hyperglycemia is finding way too much glycine in the cerebral spinal fluid. Because that faulty cleavage system is also active in the brain, measuring the CSF to plasma glycine ratio is a really critical diagnostic test.
We've touched on this a few times, but let's really nail this down. It's glycine's relationship with one-carbon metabolism that truly cements its role as a central hub in our body's biochemistry.
So what we have is this really elegant metabolic loop. Making glycine from serine produces a one-carbon unit for the Foley pool. And breaking glycine down with the GCS also produces that same one-carbon unit. So glycine is both using and supplying this critical resource, linking amino acid metabolism directly to the folate cycle, which, as you know, is absolutely essential for making the building blocks of our DNA.
Okay, let's pull all that together. Time for a quick recap of the most important things to remember about our little multitasker, glycine.
And here's a simple mnemonic to help you lock in 3 of glycine's biggest jobs for your exams. Just remember, glycine gives creatine, collagen, and calm CNS. Creatine for energy, collagen for structure, and a calm central nervous system thanks to its neurotransmitter role. That's a great one to have in your back pocket.
All right, time to test yourself. First question, which cofactor is absolutely essential for converting serine into glycine? Think back to that one-carbon connection we talked about. The answer is tetrahydrofolate, or THF.
And one more, a clinical one this time. What's the specific enzyme system that's broken in nonketotic hyperglycemia? Remember that major breakdown pathway.
That's right, it's the glycine cleavage system, or GCS.
And that's a wrap on glycine. It's such a great lesson in biochemistry, really. It shows us how the body can take one of its simplest little building blocks and use it to create incredible complexity, from our genes to our muscles to our very thoughts. I hope this was helpful. Join me next time as we take a closer look at glycine's metabolic partner, serine.